Hello and welcome to Neuro Navigator. This audition series is composed of human ear anatomy, hair cell transduction, major auditory pathways, and signal processing. Today's topic is hair cell transduction. In the last video, we talked about the cochlea and a rough overview of the structures it holds. In this video, we will be talking about it in much greater detail because the cochlea serves as the base for auditory sensory transduction. In the cochlea, we have this spiral-like structure called the cochlear partition, which has 3 fluid-filled channels. This is represented by this drawing here. In this top section, we have the scala vestibuli, and the bottom section is called the scala tympani. Both of these sections are filled with perilymph, which has a very low potassium concentration and comes in at about 0 millivolts. The other section here, this triangle piece, is called the scala media, and it has a stratified epithelium section represented by that yellow right there, called the stria vascularis. The cells here are going to pump potassium into the endolymph, which is what the fluid is inside the scala media. Unlike the perilymph, the endolymph has an obviously high concentration of potassium, and it comes in at around 80 millivolts. But we'll set that aside for a moment and talk about the hair cell, which acts as the sensory receptor and transmits vibrational energy into electrical signals. At the very top of the hair cell, we have these things called stereocilia. The apical side, which is everything up here of the hair cell, is in scala media and in the endolymph. And the soma, or the cell body of these hair cell, is in the perilymph down here. Which brings us back to this mess of a cochlea. When the basilar membrane vibrates, it moves the tip of the tallest stereocilia right here. This is linked to the shorter stereocilia with tip links. The displacement, so this is a shearing force across the top, this is already being sheared. The displacement stretches the links and opens cation selective channels. Conveniently, they are called hair cell mechanoelectrical transduction channels, or HCMET. Remember that these cells are extended between areas with opposite potassium concentrations. There is an endocochlear potential where a large electrical gradient drives potassium into the hair cell. In the soma, when you have this large amount of potassium driving this large electrical gradient, this opens voltage-gated calcium channels, which lets calcium in and helps glutamate to be released into the synaptic cleft. Voltage-gated potassium channels are then opened, allowing potassium to leave the hair cell and flow down its concentration gradient into the perilymph. When calcium flows in because of the voltage-gated calcium channels, those channels will also open, allowing potassium to leave, repolarizing the cell. If you're struggling to remember perilymph and endolymph and stria vascularis, here's the big takeaway. Potassium enters the stereocilia because of the electrical gradient and leaves the soma because of the concentration gradient. If you want to know more about how sound is transduced, follow for more.